hi everyone. I spoke, I think it's 2 years ago now that I spoke on the beginnings of our pilot project when we were first setting it up. So now we have almost all of the data, although there's still some more ongoing analysis that we're doing. And we have a number of next steps that we're pursuing. So I'm going to I'm going to quickly go over the background for those who might have forgotten since it's 2 years or who are new. This is a project that we put together in partnership with Coquitlam First Nation. And I'm sure I don't need to go as much into the background on this project as I did with members of the university community, because members of this committee are pretty well informed on the impacts that have happened over the history of Coquitlam, particularly regarding the salmon. So the Coquitlam Dam building meant that the salmon run was lost. The salmon of the Coquitlam First Nation that used to populate the river every spring were no longer able to return to the ocean. So they became a residualized population trapped in the lake. Now, there were survivors that remained in the lake, and there's a healthy population there of non-anadromous or entirely freshwater kokanee type salmon. And that, of course, has led to an attempt to restore this population. And I'm sure everyone on the committee is familiar with the Coquitlam Salmon Restoration Program that's seeking to do exactly this. And this is the nature of our partnership, was regarding what can be done to help with this restoration project. And I'm not sure if it's been announced to the committee yet, but the sockeye hatchery is now operational. So the construction's completed. They have I believe they're doing a test run of coho salmon currently, but the plan is for this fall, with the fall season, probably in October, they should be able to fish for broodstock in the lake, which can then begin the process of raising smolts in the hatchery to be released in the watershed. And hopefully this will lead to more returning fish. There have been some returning fish, either from the previous releases that have been done of fish from the lake, or through the fish valve that was constructed by BC Hydro. There's not a lot of fish movement through that valve, but there are some returners. I was talking to people at the DFO just last week that they are up to, I think they have 13 DNA samples from confirmed returning fish over the last couple of years, or from last year, which was quite a large jump from previous years. So we're hoping that that means we're going to start seeing more and more returns. Certainly once the hatchery is in operation, it'll be 4 years from the beginning of the operations before we're likely to see returners. And of course, there are several challenges to this project, not the least of which is the extensive urban development of the watershed. Again, I'm sure all of you are aware of this. Just looking at the satellite image, you can see the site of the hatchery just below the dam, which is upstream of urban development, but the lower watershed where these fish are being released and where they have to return to breed is almost entirely urbanized. And you can see the main watercourse passing past the gravel mines, industrial area, the suburbs, through downtown, and then into the outflow near the Colony Farm. And so the idea here is what kinds of toxic impacts might be occurring because of this urban development. And this is an area, I'm a developmental toxicologist, and I work with fish embryos. And so you get events like I'm sure everyone's heard of the fish kill that occurred in 2019 that wiped out a number of fry in the Hoy Creek hatchery. Coquitlam First Nation has reported a number of other localized fish kills in specific tributaries or streams and catchment areas. And that's part of what we want to combat, is to start to figure out what toxic impacts are happening in the water due to the urban development. And when it comes to urban toxicity, it's always a polychemical situation. So when you're looking at something like a local mining site, for example, on a river outflow, you know what industry it is, you know what specific chemicals might be a problem that's coming from that site. But when you're dealing with an urban environment, there is so many different chemicals that are interacting with each other that you really can't easily determine what's going on. So you get things like agricultural runoff, residential runoff, anything that goes into wastewater treatment. People think wastewater treatment removes all the chemicals. It doesn't. All it does is kills the bacteria. It's just chlorine treatment. So any stable chemical that goes into household wastewater remains in the water that comes out of the sewage treatment. And then, of course, anything that's used in the yard or even on people's bodies that washes off goes into urban runoff and ends up in the water. And so all of our catchments are picking up the detritus of whatever we're doing in those areas. So this leads to sort of what I call the central problem of toxicology. Whenever I teach students about aquatic toxicology, I like to show them these papers or these figures from this one paper, just to really illustrate. This is a graph since the 1970s of tracking the synthetic chemical output of different industries. And you can see this is extremely exponential, right? Pesticide use, chemical industry, outputs, pharmaceutical consumption, and so on. And when you look on the right, you see the publication rates of different environmental topics. And if you look specifically at the synthetic chemicals, you can see that it's basically flatlined. It hasn't really increased in decades. So the bottom line is we are producing new synthetic chemicals way faster than we can possibly study them. And that leads to the approach that is starting to become more common and that we were pursuing, which is to look at generalized measurements of toxicity. So one way to discover what are the contaminants of emerging concern and how are they working together is to take all the chemicals from a given water sample, either by concentrating that sample or by removing the chemicals by some kind of a process, and then testing them all together in a model like the fish embryo system, which is what my lab does. So here you can see an example of this from a study in the Danube River, where you can see concentrations by relative enrichment. So simply how many times has this been concentrated compared to the water coming straight out of the river, and then measuring the fish embryo test toxicity so that if the concentration goes down, you know it's more toxic. And so you have a site here from this particular location along the river where the toxicity is much higher than the average at other sites. And even without knowing what chemicals are in there, you can start to do remediation. So things like bioremediation, like building things like rain gardens at targeted sites where you know that there's something going on, even if you don't know what, is a strategy that municipalities around the world are starting to use. And I've actually been talking with some people from the Ministry of Forests about this exact strategy. That's now the strategy that's happening in BC, is to look at rewilding and bioremediation using plants and soil embankment and other means to filter catchment and water flow. The other side of what we're looking at is the environmental DNA that's present in a given water sample. And environmental DNA is basically the idea is that our ability to detect DNA has gotten so good that we can actually measure specific details of residual DNA in an environment produced by the organisms that live there. So most of these organisms are microbes, and they're producing bacterial DNA and fungal DNA, but also the larger organisms like the fish that live there produce DNA. And one of the ideas that we had was, can we look at the bacterial community present in a given sample and see if there are changes that are happening due to the specific activities at that site? So any anthropogenic activity has an influence on the environmental DNA from a given sample, right? Whatever's going on in terms of industry or any of these things that I talked about in terms of urban impacts will affect those bacteria in some way and other organisms that are there. So this is the two sides of the approach that we took with our pilot project. So this led to the formation of a pilot project we started in the fall of 2023. Coquitlam First Nation helped us identify 5 sites to start with, so a very modest pilot. Our site 1 was chosen upstream of all of the urban developments. So near the hatchery site, the only urban development above that in the stream is the dam itself. But we couldn't go any further up without a lot more difficulty. So that was the first site. Then we chose a couple sites on the edge of town, one within the gravel mining area, one at the edge of the suburbs. Then we had a site in downtown Port Coquitlam where the water access was easy. And all of these sites were chosen in part because of the ease of access in order to get water samples. And then finally, we chose a site in the outflow. And in the end, we decided to use that site mostly as a positive control because we actually chose a site that's within the tide effect boundary. So there's actually a lot of salt in the water there based on the tides. And so when you concentrate that to look for toxicity, we actually expected to see toxicity because of the salts. So that became a pretty good positive control for us. There we go. So basically the strategy for this project was we had students from the KPU Environmental Protection Technology Program who are trained in soil and water sampling. And we used their coursework where they do capstone projects to go out and take water samples from these sites along with the KFN Water Guardians. And then once we had those samples collected, we did chemical and physical testing of the samples, some of them on site, some of them back at the lab. And then we had water samples for eDNA extraction, where the water is filtered through a filter unit. And then we do a 16S, which is a gene that all of the bacteria have. And different species will have different DNA sequences for that gene. So we could then do sequencing to look at what's going on with the overall biome, the overall microbiome or microbial community of bacteria in that sample. And then we also took large volume samples that we could then concentrate with an evaporation cabinet or a desiccation cabinet to bring them down to high concentrations that we could then use to expose to fish embryos. And the fish embryo test is a highly standardized toxicity test, which allows us to measure both the acute toxicity or lethality and developmental toxicity. And so we did this, and now I'm going to tell you about the results of the project. So hopefully that brings everybody up to speed on the project that we theorized last time, but now we have actually achieved. So here are some of the students at KPU doing toxin exposures with the fish embryos and doing genome sequencing and bioinformatic analysis. So I'm not going to go through all the data because there's a lot. Most of our physical and chemical testing data was not especially interesting. There were a lot of things we tested for that we didn't see anything worrying, which is good news. We did see a lot of variation in turbidity. The highest turbidity tended to be at the mining sites, which is not too surprising, where the gravel mining is going to create more silt in the water. We also saw some pretty interesting events, so that we had 7 separate collection dates, as you can see. And you can also see this one date, which is during storms, where we actually saw really high turbidity upstream that came down as we went, which tells us there's a lot of dust and silt that gets into the catchments during a storm event. But even within these, only at the highest upstream site near the dam did we see anything worrying. Drinking water standards, even for turbidity, is generally between 1 to 2. And obviously this is stream water. It's not really intended, you don't think of it as being as clear as processed drinking water. So these numbers aren't particularly worrying. What we did see that was worrying is these transient pH spikes, which I've highlighted. And so these were tested on site, and we only ever saw this at sites 3 and 4, which are within the suburbs and downtown Port Coquitlam. And these are likely associated with individual discrete dumping events. One of the things that has been theorized about these fish kills is that they may be related to people getting rid of pool water that has chlorine, or in the case of these alkali spikes, detergents. So things like car washing or driveway washing, where someone is rinsing detergents and they're going into the catchment, and you get a discrete event. Often detergents are associated with very high pH, so this may be involved with some of the fish kills that are being witnessed. I'll come back to that because that's related to where we're kind of going next with this project. With metal analysis, we saw high metal contamination for aluminum. Interestingly, we saw this only in the fall, first 3 samples, and when we were looking in the spring, these numbers came down. And also, it was very interesting to us that we saw this at Site 1, which is above the urban development and industrial sites, which tells us that some of this contamination might be coming from the concrete of the dam itself. There is some research to suggest that metal leaching can occur with large sources of concrete. Aluminum is one of those metals. In order to test that, we would need to get water samples from Watershed above the dam, either in side tributaries where the dam isn't being affected, or above the dam itself from the lake or elsewhere, and then compare that to the downstream. After the fall set of samples, when a different set of students came in, we did some additional metal analysis. So where we were seeing no interesting changes, we went and looked for some other metals, and one that we saw consistently was high copper. A couple of sites where lead was near the threshold for BC water quality guidelines, but more consistent was the very high copper, which you can see is actually 10 times above 10 or 5 to 10 times above recommended levels. So that's all I'll get into here. There was a lot of other stuff that we looked at, but these were the ones that were particularly interesting. Moving on to the toxicology work, I've got all of that data summarized here except for the last collection, which I just had a student finish this like 2 weeks ago, so I don't have figures yet. And I won't go through all of this. I'll walk through the greatest hits here to make it clear. But what you can see is the increasing concentration. This is relative enrichment. So concentration up to 1,000 times, right? So 1,000th of the volume of the original water sample. And as you can see, we always saw Site 5 with nearly complete lethality, 0% survival, which was expected because of the salts. But at a number of collections, we also saw Site 4 with high lethality at the highest concentration examined. And in some cases over in C, you can see Site 3. Can you guys see my arrow with my mouse? Okay, I might try and turn on, I think I have a laser pointer function. There we go, that's better. So you can see Site 3, but not 4 in this particular collection date was highly toxic, or in many cases, both. So here you can see both Sites 3 and 4, and even Site 2, quite significant reduction in survival. This particular sampling date was the storm date, and that's where we actually lost some samples because we weren't able to collect them. It turned into a hailstorm that day. And I've summarized all of that data here, so you can actually see, just looking at the highest concentration from these different sampling dates, you can see where the lethality or the survival comes way down. Site 5, which is expected, but also Site 4, which I'll remind you is downtown Port Coquitlam, and Site 3, occasionally worrying as well, and that's where the suburbs first start to affect the catchment going into the river. So this tells us that there are, at least on certain discrete dates, effects that are causing fairly significant increases in acute toxicity. That said, this is at only at the highest concentration we looked at, 1,000 REF, which is quite high. So it's not, you know, oh no, we can't go swimming, it's going to be crazy toxic. It just tells us that it's more toxic than upstream sites, and that there may be something in there that we want to do further work with. In terms of developmental toxicity, we only saw any developmental toxicity at Site 4 and only on certain collection dates. So these are zebrafish embryos. Along the top row here, you can see A is an untreated with just regular water. B and C, you can actually see a heart edema, and that's a fluid retention within the developing heart that's likely to lead to problems that could lead to lethality. But you can see that we actually saw this effect even at relatively low concentrations on certain dates. And in particular, this collection here in the light blue is one of the collections where we saw a spike in pH at that site. On rare occasion, in the bottom row here, you can see a very severely affected embryo with no head. We only saw that once in each of these two collections. So the two embryos are shown here, which means we don't have enough numbers to make any kind of analysis or predictions on that. So that's something we'll consider later. The arrow here just shows that we had 100% lethality for this particular group. So we actually couldn't measure any edema because they were all dead. So that doesn't mean they weren't affected. It just means that there's no numbers to analyze. So that's all of our toxicity data. Then we went looking into our eDNA. Oh, I forgot I put this slide in. So just before I get into the eDNA, we tried to look at some correlation of some of this data. Now, because this was student projects, we didn't always get toxicity samples and chemical analysis samples from the same dates. The students had to like pick and choose what they were collecting. So some of these just don't line up, but for these first 4 dates, these are dates where we had collection for both toxicity testing and for chemical testing. And you can see that some of this lines up where you can see that we have pH, like particularly that second collection from site 4, we had the aluminum that was high throughout, but also the pH spike, and we saw acute toxicity and heart edema, developmental toxicity, all from that one sample. We also saw a spike in acute toxicity at this site with the pH spike. We didn't see that here, but the pH spike was upstream of where we saw heart edema forming. So there are some probable connections between these thresholds of toxicity and the chemical events that are occurring. So this is all from the pilot. Obviously, there's lots more to do, and I'll talk about that in a second. With the eDNA, we actually saw some very good news, but also very interesting proof of concept. So when we did the bioinformatics analysis looking for dissimilarity in the microbial community, and so this accounts for both changes in species and changes in abundance, we found that they clustered not by site. On the right-hand side here, you can see that the individual color-coded sites are kind of all over the place. There was no consistency, meaning that the bacterial community differences weren't really significant from site to site, but they were from season to season. So when we looked at all of our fall, winter collections, we saw a cluster of similarity within the bacterial species. And when we looked at our spring and summer, we saw a slightly less similar, but still clustered area. So that's good news in that it shows that whatever toxicity might be happening at some of these sites, like site 4, isn't sufficient to cause a lot of problems for the overall microbial community or a lot of changes. Seasonal changes are to be expected. When the temperature is greatly different, we would expect different bacteria to thrive at those temperatures. So this was actually some relative good news. When we broke it down by time period, we did see some differences. So when we looked in the colder months specifically, we did see that site 4 clustered over by itself. So there were some differences in the bacterial complement within site 4 compared to the other sites in the winter, but not in the summer. In the hotter months, we saw pretty random distribution of different species. One thing I found surprising was even at site 5, where there's higher salt, the bacteria didn't care. It didn't significantly change the bacterial community that we were seeing. And then we were looking also for abundance, because in particular, we were trying to see if there was any changes in abundance of bacteria relevant to the salmon, whether those are pathogenic bacteria or beneficial bacteria. And what we saw was there were some differences in certain taxa. So each of the dots, and I'll walk you through this as it's a complicated figure, but each of the dots here represent one taxonomical group, whether it's a species or a genus or group of bacteria. And what we're looking at is relative abundance, and the comparison at 0 is the hot weather months. So you can see that in the cold weather months, there is increased abundance or decreased abundance, depending on which way you go, more or less. And we analyzed these for statistical significance, and we only saw a few taxa where there were significant differences. So up here you can see 3 taxa that were considerably less common in the winter, and 1 that was considerably more common in the winter. None of these taxa were salmon specific. None of them were pathogenic. So that's, again, good news. We weren't seeing changes in bacteria related to, or that might have an impact on the salmon. And so one of our next steps is to dig deeper into this. We have a list of specific bacteria that are either pathogenic or beneficial or neutral to the salmon. And we want to look at abundance. Since we're not seeing differences in abundance by site, we want to just look at abundance within the urban area compared to references from other watersheds. So in other words, are there worrying levels of salmon pathogens compared to a non-urban watershed, for example? And that analysis is still ongoing. So to summarize all of our results, we saw seasonal changes in metal contaminants and seasonal changes in the bacterial community, which is expected. The seasonal changes in metal contaminants was not expected, and that may represent something going on with concrete effects in warmer or colder months. We saw a lot of site-specific toxicity, particularly in downtown Port Coquitlam. That's not too surprising, because downtown is where we're going to see a lot of the cumulative effects. But now that we have that proof of concept, we can start looking at more specific areas and tributaries, which is really going to be interesting. The sites did show transient water quality changes, which tells us that there is likely discrete events like dumping that have pretty significant effects on the toxicity. And so this gives us proof of concept both for our methodology for looking at general toxicity in concentrated samples, but also for measuring eDNA by specific sites and dates, and getting robust quantifiable data from that eDNA. And so this leads me to the last section, where I want to talk about our future directions. One of the major limitations for our general toxicity is that using a desiccation cabinet, and you can see a desiccation cabinet set up here, where dehumidifiers, industrial dehumidifiers, are being used in a sealed cabinet to dry these 20-liter samples. It takes 2 to 3 weeks per sample. And what that means is that any unstable chemicals or volatile chemicals are going to be lost. So you're only looking at a subset of the chemicals, and there may be effects that we're not seeing, toxic effects in the water, just because their chemicals aren't being collected. And you can see at the extreme concentration, it looks pretty nasty. But even at these high concentrations, we weren't always seeing toxicity, which is interesting. So that leads us to what we want to do next here. We want to really move into solid phase extraction. And Coquitlam First Nation has put in a grant that we've partnered with them on with the Canada Water Agency to get equipment that you can use for solid phase extraction. And the idea here is that you can pump very large quantities of water through these solid phase cartridges. And what's in there is adsorbent that will actually absorb all of the chemicals in the sample. And so you can run like 100 liters through a sample, collect all those chemicals, and then you take them to the lab and you elute them, and you get much more rapid dry down. And so you get way more volume for concentrated samples and way more quickly. And that will allow us to do a lot more sites and a lot more testing. And in particular, that sort of testing allows us to do more advanced bioassays with the fish embryos. So we can look at specific things like not only developmental toxicity, but genotoxicity, hormonal toxicity, and so on. And then, of course, if you have larger volumes of samples, you can fractionate them and find the more toxic fractions of chemical, which means you can start looking to try and identify what contaminants are in there that's causing the toxicity, which obviously is very valuable information to us. And so when we have the ability to look at more samples and more quickly, obviously we can start looking at more sites. And so we're actually starting a second pilot this summer. I received some funding from the Indigenous Services Office at Coquitlam University to hire some students to do some more sampling using the methods we're already using. But KFN is going to help us pick some specific tributaries in order to look at sites of concern. And then the last thing I just want to quickly talk about is the future side project that's growing out of this, which is to look at the genomics of the salmon themselves. Now that we have methodology for eDNA that's working very well, we can actually start looking at applying that eDNA technology to the salmon in the watershed, not just the bacteria. And so one of the things that we're doing right now with the Applied Genomics Center at KPU is to do some genome sequencing of the salmon. So we have access now to individuals from the lake population, and we'll have access to more that are collected this fall. But also the returning anadromous fish samples from the DFO. They're willing to share those with us to get DNA sequencing. And then also outgoing smolts that BC Hydro is tracking from the fish valves. And the idea with this partnership is to do some variability analysis. And we have some funds already to go towards this from the Makeway Foundation, which we've used to leverage funds from within the Applied Genomics Center from NSERC. And we have partners now at DFO, Tim Healy and Eric Rondo at Pacific Station are really interested in looking at the differences in DNA between the lake fish and the returning fish. And so is BC Hydro, of course, Coquitlam First Nation. And one of the things that we're looking for is Watershed Society volunteers. We've already talked a bit with, I've talked a bit with Georgia, and we've talked a bit with the Hoy Scott, or the Hoy Creek Hatchery and the Hoy Scott Watershed Society. But having volunteers to help our EPT students get to sites and collect water samples, help them move equipment, would be really great to enable us to do more sampling and more testing. And then the end goal of the genomics is going to be variability analysis to hopefully find markers that associate with successful return to anadromy. And that will greatly allow us to increase the success of the hatchery project. And then, of course, eDNA analysis with these markers could be used to detect the different species that are in the river and monitor the success of salmon abundance within the watershed. And that's everything I had to talk to you about today.